Hello, and welcome to the Foundation Fighting Blindness's first CRB1 Forum. I am Judy Taylor, Chief Development Officer at the Foundation, and we appreciate you joining us today. Before we get started, I'd like to briefly review a few details for the call. Currently, all participant lines are in listen-only mode with no video. Today's conference is being recorded and is available with closed captioning. To activate the closed captioning, please select the live transcript option located at the bottom of the Zoom interface, then select Show Subtitles. Please note that on today's call, our speakers do have video live. However, all of their comments will be provided verbally and there are no slides. If you are using a screen reader, please be aware that the controls are at the bottom of the Zoom interface. This control bar may collapse when it is not in use. If you prefer to prevent the controls from auto-hiding, go to Settings within the Zoom platform, select Accessibility, and then select Always Show Meeting Controls. It might be helpful to maximize your window and navigate by using the Tab key. Additional buttons and settings are available by pressing Alt key. During the call, you may ask questions through the Q&A and chat features, and we will address the questions towards the end of the call during the Q&A session. Now, I'd like to turn the call over to Laura Fietta. Hi, Laura.
Hello, hi. Hello, everyone, and welcome. Good morning, good afternoon, or good evening, depending on where you are in the world. I am Laura Fietta, and I'm so happy to be with you today. It's fantastic that we've come together from all corners of the world to address CRB1. I serve as the Victory for Vision Campaign's International Chair for the Foundation Fighting Blindness. Like many of you here, I'm incredibly excited to learn about advances that move us towards treatments and cures for inherited retinal diseases like CRB1. My 15-year-old daughter has CRB1 retinal dystrophy. We live in London in the UK. She does a good job of living with it at school and outside school, but it's hard. We want to stop this disease in its tracks. Better still, reverse it. Since her diagnosis 9 years ago, the research landscape has advanced dramatically. The science has really taken off. The scientists know what to do to reach their goal of treating and curing inherited retinal disease. They have clear strategies for developing treatments and cures. I have found the Foundation Fighting Blindness to be passionately international in their reach and purpose, and a fountain of knowledge on research to tackle CRB1. Both CRB1 gene-specific research and gene-agnostic research. I have found them to be in a unique position to fund, coordinate, and collaborate, working with scientists, commerce, charities, and patients around the world. They have even joined forces with Chapel in York. It's an international philanthropy agency, and it enables donors outside the US to take advantage of their domestic tax benefits when making donations. That makes a huge difference to increasing the sums raised. A vital component in this international response is FFB's world leadership in diagnostics. And of course, a key part of this is FFB's My Retina Tracker registry. It's completely free to join and it's set up for patients to use. It is the global patient registry for people with retinal dystrophy. And years ago, FFB helped my family join. Forums like the one we are having today provide us with the opportunity to hear the efforts of the Foundation and the remarkable research to combat blinding diseases. Here today, we have CRB1 affected people from all over the world, and FFB is supporting scientists around the world. It's a truly global CRB1 community, and this is how to get things done. As with the COVID pandemic, we need to pool resources for a coordinated global response in order to end retinal disease. The Foundation Fighting Blindness is leading this worldwide effort. All of us connected with CRB1, the patients, the families, the scientists, we all need to come together and pull together. If you know anyone affected by CRB1 or working in CRB1 who isn't here today, please encourage them to get in touch. There are tens of thousands of people affected by CRB1 worldwide. And if we come together, we can find treatments and cures far quicker by supporting the science and having our data ready for the science. And FFB is here pushing that science forwards, always pressing to go faster with our support. Now I'm thrilled to introduce you to Ben Shaberman, Vice President of Science Communications. Many of you know Ben from his illuminating podcast series and from the Foundation's excellent vision webinars, which help us all to understand this incredibly complex subject. In essence, he is an FFB celebrity and we owe him so much. And we are so happy to have Angela Bowman with us today as Science Advocacy Lead. She is the newest member of the Foundation's science team. You will hear about Angela's role in a moment. Speaking on behalf of the CRB1 community, we are delighted to have her and we are excited for what the future will hold with Angela on board. Over to you both.
Well, thank you, Laura, for that introduction. And thanks to you and the many patients, family members, and other constituents who have joined us today for this webinar on CRB1. We've had more than 200 people from around the world register for this webinar, which is really exciting for us. And we hope you can take away some great information from our discussions. So I think, as many of you know, there are some challenges in addressing CRB1-associated retinal disease. And one of those challenges is that it can cause a range of phenotypes. And when I say phenotypes, I mean that the mutations in the gene affect the retina in different ways for different people. For some people, it can cause LCA when the gene's mutated. Others, it can cause RP. And that's in part due to the fact that there are really 3 different forms of protein that the CRB1 gene can express. Genes express proteins. And again, in the case of CRB1, there are 3. We label those A, B, and C. And some mutations affect expression of the A protein. Others affect expression of the B protein. And again, depending on what protein is defective, it can cause different types of vision loss, different diseases. And another challenge is that our animal models, the current animal models that we have, don't mimic the human disease, the human manifestation very well. And as if that wasn't enough in terms of challenges, CRB1 is a bigger gene, which makes it difficult to deliver from a gene therapy standpoint. It's rather large for the viral containers that are used in gene therapy delivery. But thanks to research that we've funded and other organizations have funded throughout the years, we have learned a lot about CRB1, many of the things that I just mentioned, and we're poised well to learn so much more. And while, of course, this webinar, and we are strongly focused on CRB1 specific therapies, please know that there are other gene agnostic therapies in development that might benefit people with CRB1 mutations. Some of these are already in the clinic. For example, Nacuity's antioxidant, which is an oral medication, that's in a phase 1/2 trial. We have optogenetic therapies, which are for advanced retinal diseases, for advanced vision loss. They're in clinical trials. And then another company that we're funding called Sparing Vision is getting very close to launching a clinical trial for their cone preserving therapy. So a lot of good, as we say, shots on goal for some gene agnostic approaches that might benefit people with CRB1 mutations. Now, you've already heard about our strong CRB1 focus and some of the work we've done. And I'm excited to introduce Angela Bowman, who is a PhD, and she is the Foundation's lead science advocate. And she joined us on August 1st. And as you already learned from Laura, she has a specific focus on CRB1 and 2 other genes. And as my late grandmother used to say, she's a very smart cookie. I've really enjoyed working with Angela since she joined a few months ago. And so, Angela, welcome to the webinar. Thanks again for focusing on CRB1 research. And to start off, I just wanted to ask you to describe your background and why you think it's prepared you for this role in advocating for patients with CRB1 mutations.
Sure, Ben, I'd be happy to. And thank you to all of you who've joined the call. I'm so excited to engage with all of you, and I'm so excited about this role at the foundation. So I've always been interested in science, always as a kid. I completed my undergraduate degree at the University of Pennsylvania in biology, and then I went to Stanford for my PhD in developmental biology. That's the science of how we turn from a single fertilized egg into a functioning human being with arms and legs and eyes in the right place. And during my PhD, I was working on stem cells in the mouse brain. And towards the end of my PhD, my advisor suggested that I also start working on the eye. Well, I just fell in love with the eye in a way that I never did with the brain. I thought it was the most beautiful tissue I'd ever worked in. The layered structure of the cells, it just resonated with me. And I guess it's how some people like jazz and some people like polka. For me, the retina was just that, it just did it for me. So after I completed my PhD, I knew that I wanted to do a postdoc in retinal gene therapy. And luckily I found a home with John Flannery and Dave Schaefer at Berkeley. And my project there was focusing on using AAV to deliver genome editing proteins directly to the eye to see if we could correct mutations in a gene called CEP290, which also causes LCA. And actually, John was funded by the Foundation Fighting Blindness while I was there, and he talked about it a lot. And so that was my first introduction to the foundation. My postdoc was cut a little short when my husband's job took us to St. Louis, but that allowed me to transition into an administrative role, which is actually what I was looking for. I knew that I'd like thinking about science and reading about it and talking about it, but I didn't want to be the one doing it anymore. But I still wanted to be close to science to use all my training. For 8 years, I served as the executive director of the Center of Regenerative Medicine at Washington University in St. Louis. In this role, I led the programs and initiatives of the center, which had more than 100 different labs working on regenerative medicine by the time I left. This allowed me to get exposed to stem cells and gene therapy, organ regeneration studies, all kinds of regenerative medicine. I also got engaged with the Undiagnosed Diseases Network while I was there, which is a government-funded program that tries to find genetic diagnoses for people who have rare diseases. Many of them have been trying to find answers for years. And so this was a really rewarding project, and it allowed me to more fully appreciate the patient perspective in medicine, and also to gain a deeper understanding of human genetics and the factors we need to consider when developing therapies, especially for rarer diseases. When my husband's job moved us to Philadelphia, I knew it was a good time for a change, and I always thought that I would like to get back to working in the retina. This position at the foundation opened up, and it seemed like a great fit that really drew on so much of my experience. And I knew the foundation had a great reputation of being driven by science and by patients. So it seemed just like a great organization to be a part of. In addition to my professional interest in the retina, I also have a personal connection. My husband has Stargardt disease, and he was diagnosed when he was 8, and it wasn't until 3 years ago that he finally got a genetic diagnosis. And even with all my scientific training, I still found that genetic test report to be very confusing and overwhelming, which made me think how much harder it must be for people who have less scientific training than I do. And so it led me to feel this sense of compassion and really a desire to help people to understand their genetic diseases. So while I don't know what it's like to be personally affected with an inherited retinal disease, I do know what it's like to see someone you love deal with one, and to also desperately want to find ways to help them. I'm so excited about this new role, and that I'll have more time to think about science and to engage with all of you and to move the needle forward for IRD research.
Well, thanks, Angela, for covering your background. I have definitely observed that you bring a lot of passion to the science and the patient journey, and we appreciate the fact that you bring that to your role. And just for the record, I prefer jazz over polka, but I did listen to a lot of polka growing up involuntarily. So just for the record. So I've already mentioned that you're focused on driving research for CRB1. Can you talk more specifically about what that means, what you'll be doing in your role?
Sure, yeah. So the title Science Advocacy Lead might not give away exactly what I'm doing. This is a new role for the foundation, something they're exploring. Briefly, it means that I'll be a bridge between researchers, companies, and affected individuals. I'll be immersing myself in the science of CRB1 and identifying gaps so that when we as a foundation fund research, we're funding the most impactful work. I'll be the person at the foundation who can dedicate all of her mental energy just to thinking about CRB1 and a couple other genes. So for the past 3 months, I've been meeting with researchers working on CRB1, reading lots of papers and talking with constituents like you. One of my big goals moving forward is to evaluate the CRB1 landscape, to create a roadmap, to understand what milestones we need to hit and how we can get CRB1 therapies to the clinic.
Great, great. So I mentioned very briefly some of the challenges that CRB1 mutations bring to therapy development. Can you talk a little more about some of those challenges and what you're excited about in terms of addressing those challenges and moving forward, either from a model standpoint or a potential therapy standpoint?
Yeah. So yeah, as you said, there are some challenges with CRB1, and you already highlighted the fact that CRB1 is too big to fit in AAV. So many gene therapy strategies use AAV to deliver a normal copy of the defective gene to the eye, but CRB1 doesn't fit. It's like trying to overpack your suitcase and not being able to quite zip it closed, like, you know, your sweater arm is hanging out. So that's not good. But there are strategies to get around this. So we can think about using a bigger suitcase to deliver AAV or to deliver CRB1, a different kind of virus, for example. Or we could pack half of CRB1 into one suitcase and half of CRB1 into a different suitcase, and then just combine it when it gets into the cell. So we and other researchers are thinking carefully about strategies to overcome the fact that CRB1 is too big for AAV. We can also use approaches that don't rely on replacing all of CRB1, but just rely on fixing the genetic typo that's causing the disease. And this is a strategy often referred to as genome editing. So it's like a little molecular cut and paste that, you know, goes and finds the mutation, cuts it out, and puts the correct letter back in. And that, I think, is such an elegant strategy. I personally, as a scientist, love that approach. I would love it if we could find a personalized strategy like that for everybody, but the route to the clinic on that is a little more challenging than some other strategies. And then, of course, there are, as Ben alluded to, there are other strategies that don't rely on fixing CRB1 directly, but address the underlying cell death in the retina, and we call these gene agnostic approaches. And many of them are, as Ben said, in clinical trials and are showing great promise. So I think there's lots of avenues that we can explore, and it's up to us to promote all the ones that we think have a shot on goal.
Great. Thanks for that overview. And I presume, Angela, if people have questions for you about their specific situation or some of the research going on, they can reach out to you.
Absolutely. I love talking to constituents about science and the retina. I see it as a giant puzzle, and I've always loved puzzles. And so I would welcome emails from folks if you have questions or just want to chat. I would love that.
Great. Well, thank you. And we look forward to your work advocating for CRB1 patients and research to overcome the retinal diseases. So now it is my pleasure to hand things over to another smart cookie, our colleague, Amy Laster. Dr. Laster is the Senior Vice President of Science Strategies and Awards. The bottom line is she manages our grants program, which is a pretty big lift, but she does it really well. So Amy, take it away.
Thank you, Ben. Just checking. Can everyone hear me? Great. So good day, everyone. I am really glad to be here with you today. I've been asked to provide some highlights from the animal model landscape for IRD research generally, and specifically for CRB1 research. Now, I just want to kind of start at the top and sort of work our way down, or some others might see it the other way. But the first stage of translational research, meaning going from the laboratory to a clinical trial, is often carried out using animal models and cell-based models, both that I'll talk about more. Now, to study CRB1-related retinal dystrophies, several research models have been created, characterized, and used, and many of which that have been used or developed in foundation-funded research projects that advance these studies. And we are very committed to continuing to support research advancement for CRB1-related retinal dystrophy. So let's talk about these animal models. And I'll start small to large, and I will give the caveat that there are a lot of species. I won't go through all of them today, but I'm really going to hit the highlights of some that are relevant for our discussion today. So modeling IRDs in general using what we call cell-based models is becoming a very common research tool. And specifically retinal organoids, which many of you may have heard, which are developed from induced pluripotent stem cells from IRD patients, is a really exciting field of research. These potentially allow the study of the effect of the exact mutation or variant that's present in the patient. However, they do not yet fully recapitulate the specific environment within the retina of a living animal or human, and they can be quite variable from one retinal organoid to another. However, despite these challenges, there remain a lot of interest and promise in using retinal organoids. Now, in CRB1 studies, using patient-specific retinal organoids, we do expect that they will continue to help us learn more about the development process of disease, and specifically the effect of different CRB1 mutations on development during disease. Now let's move up a little bit in size. The utility of mouse models, which probably many of you have heard about, with their very well-developed genetic backgrounds, their similarity to human anatomy and physiology, it's very valuable in really furthering our understanding of human disease, as evident by the vast number of research findings realized in mouse models. So mice models of human disease, they really provide a model system for analyzing defects in comparable human disorders like CRB1 and for testing therapeutic strategies. And so they provide this really reproducible experimental system for understanding normal development and function of cells. For CRB1, there are 3 mouse models that's most prevalent in the literature to date. So there's a naturally occurring RD8 mouse model, which has a slow degeneration, and a CRB1 knockout, which removes the CRB1 protein completely and leads to photoreceptor disorganization. And then there's a CRB1 null, which basically it doesn't produce functional CRB1 protein. And overall, and Ben has said this in his opening remarks, the disease phenotype in these animals are mild and they really don't recapitulate human disease. One little nuance that I just kind of want to bring into this conversation, in mouse, the CRB proteins, they kind of localize in an opposite manner to humans. So CRB 2 protein, if you will, or CRB2 protein, it localizes in the same cells as a human CRB1. And then mouse CRB1 localize in the same cells as human CRB2. So this is a species-related distinction in how CRB1 and CRB2 are expressed during retinal development that might explain some of the reasons that mice really fail to have a strong retinal phenotype. There have been newer CRB1 mouse models generated to kind of differentiate the roles of the CRB1 proteins that Ben spoke about, specifically the CRB1A and the CRB1B, or what happens when you totally lose the CRB1B type protein, which is a newly kind of discovered CRB1 protein. In these cases, removing the CRB1 isoform, it too shows a mild phenotype. So there are studies that are ongoing to understand these nuances in expression of proteins and how disease is caused by their disruption. Now let's move on a little larger still. Now, studies utilizing large animal models of inherited retinal degeneration have proven important in not only the development of translational therapies, but really in improving our understanding of disease mechanisms. For example, the first proof of concept gene augmentation therapy that eventually led to the FDA-approved product Luxturna for the treatment of LCA due to RPE65 mutations was performed in a dog model, one type of large animal. Now, the dog is the predominant species utilized because there are spontaneous IRDs that's common in the pet population. And typically, these are retinitis pigmentosa or LCA. Cats are another source of spontaneous IRDs. And then there are some other large animal models with spontaneous IRDs or inherited retinal degenerations. And these include sheep, horse, and non-human primates. However, there are no known spontaneous CRB1 disease in these animals. And this is true for several IRDs. And so researchers use tools to produce or engineer disease in large animal species. Another type of large animal species that this is often used for is pig models. Now, the pig has proven valuable due to the ease in which the animal can be engineered or a genetic disease can be created within the pig. There are several IRD models that have been generated in pig, and there are researchers currently interested in exploring the creation of a CRB1 gene-edited pig line. We are currently supporting a research project to compare pig versus human CRB1 expression in the retina, something that has not been done before. And to date, they have shown that there are many isoforms in pig retina, as is the case in humans and other species, including the isoforms or the CRB1A and CRB1B proteins. With CRB1 protein being the most prevalent, as it is thought to be in humans. And so the team is continuing to analyze the expression profile of CRB1 in the pig, and this would inform characterization studies in a pig line. However, before researchers get to this point, they'll need to successfully generate a pig model. And in talking with them, it's anticipated that once they begin this process, it'll take probably a couple of years to generate, to characterize, and begin expanding a colony of pigs for use in understanding CRB disease and evaluating potential treatments for CRB1 in this particular animal model. So large animal models which offer important advantages over the commonly used laboratory mouse models for several reasons, namely the eye size and dimension are more like those of humans. And importantly, they have this retinal region of high cone density, denser photoreceptors kind of packed in there. Mice, they don't have this retinal region, and that can be important for a lot of IRD studies. Now, the disadvantage of using large animal models really tend to be the cost. For example, to generate, characterize, and begin expanding, say, a colony of pigs, it's about $500,000. And that's about almost twice what it would be to do a similar study or to create a mouse model. Now, other disadvantages include just generating sufficient animal models because they have a slower reproduction. And because of the longer lifespan compared to mice, the disease course in and of itself may be longer, and therefore the time to study the animal might be much longer than, say, in a mouse model. Now, no matter the model species, there is not a guarantee that engineering a disease will recapitulate human disease. However, as researchers learn more about CRB1 from Drosophila, zebrafish, mice, and of course patients, there is reason to believe that a large animal model of CRB1 disease is possible. So I just want in sum to wrap up, and hopefully in this very high level kind of overview that I provided you with the importance of animal research to understand disease, develop therapeutics, as well as the potential of having animal models that inform our understanding of CRB1 disease and future therapeutic options. And so with that, I will stop and turn it back over to our moderator.
Hi, Jason. This is Judy Taylor. Jason, I'm going to hand it over to you to handle the question and answer session and introduce the folks who will be presenting or answering questions.
Awesome. Well, thank you so much, Judy and Amy and Angela and Ben Shaberman, and all of you for joining us today. This is a really important call for us, a really important webinar for us here at the Foundation Fighting Blindness. My name is Jason Menzo. I'm the CEO here at the Foundation, and one of our other colleagues who will be speaking here in just a few minutes, Jason Morris, mentioned it just before we started this call that this may in fact be the largest congregation of folks interested in CRB1 in one setting for one call, gathered for one purpose, really ever. And that's really exciting. There's over 100 people that are on this call right now. We're recording this call, and I have no doubt that within the couple days from now that there'll be hundreds more that'll have viewed this and digested the content that we're going to be covering today, which is terrific. I want to take a minute and just remind you, if you're looking to ask a question, there's a couple ways you can do that. Number one, you can access the Q&A and chat features here within the Zoom interface. They're along the bottom of the Zoom control bar, and you can use those tools to chat in your question, or you can use the Q&A feature. I'm gathering the questions as they're coming in, and I'll be directing them to the members of the team. I do want to take a moment, in addition to let me say this, in addition to using the chat and the Q&A, you can also email us anytime at info@fightingblindness.org. If we don't get to your question here live today, we'll certainly be following up with everyone individually in the weeks ahead. I do want to take a second, though, and recognize the team from the Foundation who is on this call. So far, you've heard from Judy Taylor, who's our Senior Vice President and Chief Development Officer. You've heard from Ben Shaberman, who's our Vice President of Scientific Communications, and our own FFB celebrity who hosts the Eye on the Cure podcast and so many other great scientific content out in the marketplace. You've heard Angela Bowman, who's new to our team, who's really specifically focused on developing strategies and plans for a couple specific targeted indications, CRB1 being one of them. In addition, you've heard from Amy Laster, our Senior Vice President of our Scientific Strategy and our Awards Program. In addition to that team, you're joined not only by myself as the CEO, but also Claire Gelfman, our Chief Scientific Officer, Peter Ginsberg, our Chief Operating Officer, Rusty Kelly, who's the Managing Director of the RD Fund, Todd Durham, who's our Senior Vice President of Clinical and Outcomes Research. And the reason I go through that is so that you as a constituent can feel how important this specific area of inherited retinal disease is to us. We've got a team of senior folks that are committed to this. We're committed not only to educating the constituency and driving the research, but we're committed to having calls like this with frequency. We envision this as the beginning of a longstanding commitment to you as a constituent here, specifically interested in CRB1. I envision bringing researchers and other subject matter experts into calls like this in the future. And as I said a few minutes ago, I want to reiterate, at any point in time, if you want to speak with anyone from the foundation, you can get in touch with us at info@fightingblindness.org. It'd be our pleasure to walk through the research with you one-on-one or to share additional content and information with you. So with that background, I now want to get to the reason that we've got time set aside, which is to answer your questions. The first question I'm going to actually direct to Claire, who's our Chief Scientific Officer. And Claire, we've talked here on this call and really in a lot of our webinars, a lot of our educational settings, we talk about gene therapy and gene-specific therapy, but we also know that gene-agnostic therapy is really important as well. Ben Shaberman mentioned a little bit about that earlier. Maybe you can talk about the difference between the two and why it's important to have multiple shots on goal.
Sure. Thank you, Jason, and welcome everyone. This is Claire Gelfman. We talked a lot about some of the complexities of what we know about the CRB1 gene. And when we talk about sort of generalized gene therapy, it's in simplest terms, it's restoring the cells with a corrected copy of the DNA so that the correct protein can be made. And that's a very specific way of thinking about a therapy, giving back a good copy, if you will, of the gene that's normally mutated so the correct protein can be made. And a lot of our efforts are focusing on just that. But we take a toolbox approach. We want to have many shots on goal. And another therapeutic strategy are what we call these mutation agnostic approaches. So these are therapies that can help restore vision or at least prevent further vision loss. That is a treatment that's not specific on knowing which specific mutation that you have. And the reason for that is that what we are capitalizing on, if you will, are the problems that happen with the photoreceptor in allowing for proper vision. And we know, for example, that different pathways go awry during photoreceptor degeneration that compromise vision. And so one of those that Ben mentioned at the beginning of the call was these antioxidant approaches that are being utilized. And there's a company called Nacuity out of Fort Worth that is leveraging just that. We know that oxidative stress is part of the etiology, part of the development of a malfunctioning retina. And so this company is testing in human clinical trials in Australia and hopefully soon in the US as well, an antioxidant approach that's taken orally in a pill. It's called NAC. It's called NACA, that stands for N-acetylcysteine amide. And it is a pill you can take that's an antioxidant that is being tested in individuals with retinitis pigmentosa who also have an Usher syndrome diagnosis. This type of agnostic approach, agnostic meaning it doesn't matter the genetic cause for your retinal degeneration, but it can help people not only with CRB1 mutations, but mutations in other of the many inherited retinal diseases as well. Ben also mentioned optogenetics. This is another mutation agnostic therapy to restore vision to individuals who have lost a lot of their vision, so later stage photoreceptor degeneration. We know that for functional vision to occur, the light that comes in through our pupils is captured by our photoreceptors, and that message then transmits to our brain so that we can interpret the image in front of us. Well, wouldn't it be nice if the photoreceptors are not functioning and cannot handle that, to give that role to a different part of the eye, a different part of the retina that's not degenerating? And that's what optogenetics is all about. It's giving that ability to sense light and understand and see the image in front of you to a region of the eye that's not degenerating. And there are a lot of companies that we are very involved with who are testing that therapeutic strategy, and that's also in later stage clinical trials. So I give those two examples of these mutation agnostic approaches, ways to think about treating photoreceptor disease that compromises vision in a way that's different from the more gene-specific approaches. And I'll just mention super briefly, cell-based therapies also fall into that agnostic approach when we are looking to restore the cellular environment of the retina in order to restore function.
Awesome. Thank you so much, Claire. That was a great explanation. And I think it's an important message for everyone on the call today that as a foundation, we not only think about one approach to treatments and cures as it relates to any condition, that we're really thinking about multiple strategies, multiple shots on goal, whether it be gene-specific therapies, including gene augmentation, genetic editing, et cetera, or gene agnostic, as Claire described today. And she described several different approaches, even within the gene agnostic arena, that could potentially have benefit for individuals affected with mutations in the CRB1 gene. So that was great. Thank you, Claire. Ben Shaberman, I want to go to you next. So there was a question that was chatted in. And for those who have not subscribed to our Eye on the Cure podcast, this is a great chance to plug the podcast that Ben Shaberman hosts, which is exceptional. It's enlightening, it's entertaining. You do a great job with that, Ben. But yesterday you had some folks on that were talking about artificial retinas, and I thought it might be a good segue to talk about artificial retinas, but also we talk about artificial retinas sometimes in the arena of cell therapy, and maybe you can speak a little bit about how those different technologies might someday play an effect on folks affected with CRB1.
Sure. Thanks, Jason. So for optogenetics and artificial retinas, what's usually most critical is that somebody have a healthy optic nerve, the cable that connects the retina to the brain, and that they have healthy ganglion cells and or bipolar cells, which survive after people have lost a lot of their rods and cones or all of their rods and cones. And those are the cells that, at least in optogenetics, are made light sensitive, as Claire was saying. And generally speaking, depending on the artificial retina, those cells are important to the function of an artificial retina. Now, different artificial retinas connect to the retina or their surviving cells in different ways. But generally speaking, again, if you have a healthy optic nerve and surviving bipolar and or ganglion cells, you should be in good shape. Now, cell therapies get a little more tricky. What we've learned with cell therapies, at least early on, is they need a healthy, really just a healthy environment to integrate into. And some of the earlier cell therapies did not integrate well with a retina that's suffering from advanced disease. And we learned that, at least with one therapy, you needed a more healthy retina for those cells to survive. So for cell therapies, it may be a little trickier depending on the person's specific retinal situation.
That's great. Thank you so much, Ben. And we have a question that was just chatted in from Thomas Collins. Hi, Thomas. About how to find Ben Shaberman's podcasts. And certainly on our website, you can search Eye on the Cure, EYE on the Cure podcast, or, which I do personally, anywhere that you get any normal, any other source to listen to podcasts. I listen to it on my iPhone in the podcast app. You just search Foundation Fighting Blindness or Eye on the Cure, and they're there. Ben, you have a new episode every other week, right? Every other week on Friday?
Generally
Just about-
speaking, every 2 weeks, and a week from yesterday, it's seen a Therapeutics, a gene therapy company, just as a
Great.
preview.
That's great. Thanks, Ben. I want to shift. So I mentioned a few minutes ago, we're joined by Todd Durham. Todd has a really important role here at the foundation, leading all of our efforts related to our clinical studies and also our MyRetina Tracker registry. Todd, I'm going to kind of package a couple different types of questions to you. One, maybe you can talk a little bit about MyRetina Tracker and how folks can get enrolled in MyRetina Tracker, how they can get our free genetic testing through MyRetina Tracker. And then also, when an individual is tested with a genetic test, what is it that it's being detected? So when we talk about different variants within CRB1, for example, what does that mean? And then finally, maybe you could talk a little bit about UniRare as well, and that announcement we made just earlier this week.
Okay, I may take those in a slightly different order.
Okay.
Let me start with what a person learns from their genetic test report. Your genetic test report will identify specific variants in your CRB1 gene, if that's the gene that's causing your condition. And those specific variants are important to researchers. Researchers will collect the information on those specific changes in your DNA. When they do a clinical workup, with a large enough collection of individuals with variants like yours and a clinical workup, they can begin to make the connection between specific variants in CRB1, variants, I mean mutations, and how those tend to appear as clinical manifestations. So this is the importance of non-interventional or observational studies like the ones that the foundation conducts, where you can see that certain variants in CRB1 are associated or tend to predict an early onset condition like LCA. Certain variants tend to predict later onset conditions like macular dystrophy or retinitis pigmentosa in CRB1. So that's the importance of the genetic test for CRB1. You obviously are the expert on your own condition, but reporting that through research helps researchers put all this information together and helps ultimately design clinical trials, because each of those clinical manifestations or phenotypes may have different strategies towards treatment and ways to design clinical trials. And that brings me to MyRetina Tracker, which is, as you know, a large study that the foundation here funds. This is a way for individuals with inherited retinal diseases to record their own genotypic information and phenotypic information. How does your condition manifest? When did you have onset of your first symptoms? And how is your health today? And this is important to us. We work with researchers all the time to collect this data so that we understand what the frequency of variants are that can help us understand the underlying prevalence of conditions like CRB1 and others. And you can join MyRetina Tracker registry at any time through our website. The main link is there. And I would also like to remind folks that if you have questions or difficulties accessing the site, we have 3 full-time staff who are ready and available to assist you. And even if you have questions after today, you can send a general email to the email address Jason mentioned at the top of this webinar, and our staff will get back to you on registering. And the last point I'll bring up that Jason asked me to speak about is a new study that we're initiating later this year, next month. This is called the UniRare study. This is a study of 1,500 individuals with inherited retinal diseases. This is the largest study of its type that we have ever funded. And it has 2 parts to it. One is to collect the information on the genotype and the phenotype for individuals with what I would say are less prevalent disease-causing genes. And CRB1 certainly would be in that category. And your investigators who are participating in this study will be recruiting their patients for the study. And this allows us to make those associations between the genotype and the phenotype and learn more about these various conditions, which is especially important for CRB1, as Jason and Amy and others and Angela mentioned at the outset. CRB1 is well known to have high degree of phenotypic variability.
Todd,
and yes.
Just for our audience, can you describe what phenotype and phenotypic means, just to spell it out?
Phenotype is really how your disease appears. It could be something like attributes of your retina, the structural part of your retina, or visual function, or the age of onset. So the difference between like an early onset condition like LCA or a later onset like RP, that's an example of a phenotype.
Thank you.
And the study also has a second part, which is a longitudinal part, which we are following particular genes, and that will allow us to follow the progression of those conditions and ultimately help design clinical trials in a way that they'll be more likely to be successful.
It's terrific. Thank you so much, Todd. And I'm keeping an eye on the clock as well as trying to monitor the questions as they come in. We'll probably have time for one or two more questions, but I want to take an opportunity, Rusty Kelly, our Managing Director of the RD Fund, to briefly talk about how the RD Fund plays a critical role in taking things from preclinical into the clinic, particularly as important technology not only approaches the clinic but moves from academia into private industry. And then anything you'd like to share about how the RD Fund may be specifically looking at opportunities for CRB1 today and in the future.
Yeah, thank you, Jason. As was alluded to earlier, the RD Fund is the venture arm of the Foundation Fighting Blindness. And the objective of this venture arm is to serve that mission of finding treatments, preventions, and cures for IRDs and dry AMD. So we have created a model that allows us to make investments, often seed investments. So these are new companies that are seeking to develop therapeutics, and often from grantees of the foundation. So it's a beautiful model that's already proving out. And we've made 12 investments thus far. 11 of those were our therapeutics, and a couple of those companies have been purchased. And it's worth noting that our mission is twofold. These are mission-related investments. We're seeking a clinical return and a financial return. The financial return is such that any return on investment goes back to the foundation so that it can further serve its mission. So with that backdrop, and as it relates to CRB1, we're working very closely with Laura, Jason Morris, many other families, many researchers, companies that have expressed interest in CRB1 to figure out what is next in the development paradigm for one of these treatment strategies, whether it be a prime medicine gene editing strategy or a gene augmentation strategy, or as Claire mentioned earlier, a gene agnostic approach. So we're taking this holistic approach to figure out how we can target CRB1 among many other indications in a variety of ways to de-risk these technologies at a very early stage.
It's terrific. Thank you so much, Rusty. It's 11:52 here on the East Coast. Probably got time for a couple final questions. I'm going to actually try to address two or three really quickly, and I'm just going to do it myself. But one thing that I want to commit to the audience is that the resources that are available at our website at fightingblindness.org are significant. We take a lot of care and put a lot of attention to curating information specifically for the patient community and the broader community. If there's ever a piece of information that you find lacking and that you would like more detailed background on, like, for example, which I had a couple of questions chatted about this, and we'll make sure that this content is created. What is the CRB1 protein, and what does the CRB1 acronym stand for, and what does that really relate to as it affects the visual process in a way that's easily digestible to a lay audience? And so that's something that we'll draft, and we'll put a primer on that on our website, and it'll be searchable like everything on our website just by typing in CRB1, for example. Second, there was a question about, just reiterating, and I saw it was chatted, but UniRare, UNI Rare, R-A-R-E, is the name of our natural history study. It's the largest natural history study we've ever conducted. I think Todd mentioned 1,500 patients that we're targeting across a variety of rare genes that affect individuals that inherit retinal disease. And then the final comment I want to make is, certainly most everyone who's here today is here for one purpose, which is not just to learn about the CRB1 condition, but more so to understand what's happening in not just from an anatomical or pathology standpoint, but really what's happening from a clinical standpoint, meaning what is being developed to bring forth a treatment or a cure for this condition. And there are a couple of folks who chatted about non-CRB1 conditions like BEST1 or other conditions that were very specific. And I want to use this opportunity to share that on our website at fightingblindness.org, we have a clinical trial pipeline tracker, where on our website, anything that's being developed or approaching the clinic anywhere in the inherited retinal disease space or dry age-related macular degeneration, it's searchable, it's updated with frequency, and that's the best resource to keep up to date on things that are approaching the clinic or that are in the clinic. So with that, we've got about 5 minutes left, and I want to turn the call over to my friend and colleague, one of our board members here at the Foundation Fighting Blindness, but also a dad of someone who's affected with CRB1, and that's Jason Morris.
Thank you, Jason Menzo, and it's wonderful to know that there's so many participants interested in CRB1 taking part in this webinar. I'd also like to thank the whole FFB staff. I hope today's conversation is the first of many. Like Laura, as Jason said, I am the parent of a child who is impacted by a CRB1 mutation. Our son, Jack, who's now 19 and is in his sophomore year at college and doing great, it was diagnosed when he was 5 years old. And by some low vision standards, his vision is pretty good, but by normal standards, it's quite poor and certainly has an impact on his daily life, but he's up to the challenges, and I'm sure, as is the case with many of you and your loved ones, doing the best he can with the hand he's dealt. As Jason mentioned, I am on the board of trustees of the Foundation Fighting Blindness. I am its treasurer and also an observer on the RD Fund board. So among non-FFB staff, I have about as close a seat to the table of what's going on within the FFB of anyone you could think of. I will tell you that the team, as you have seen here today, is phenomenal. The FFB has an unmatched reputation within the field of science, but also within the field of industry among nonprofit organizations. I have seen the results of Rusty Kelly's efforts, where $1 of FFB investment nets on average $5 of investment from for-profit organizations trying to drive research forward. And it's because they trust the expertise of the Foundation Fighting Blindness. The FFB boasts a scientific advisory board of more than 70 scientists, all of whom are experts and very knowledgeable in the field and provide valuable insight to what we do as an organization. We achieve nonprofit watchdog metrics and exceed those metrics on an annual basis, provide audited financials that are available in our annual report that comes out every year. And as has been spoken to, the My Retina tracker is a free service for genetic testing, a very expensive service that is backed by the FFB. I encourage all of you to get involved in that. Now we boast the incredibly talented Angela Bowman, who is added to our team. Could not be more excited for her role, and CRB1 is getting more attention now than it ever has at any time in history. And now is the time for us as a group to get active, recruit other family members and individuals you know impacted by CRB1 or any inherited retinal disease, and get involved. There's a lot you can do to raise money, build awareness, make sure you're on My Retina tracker and that you are counted among the people who will benefit from efforts to resolve CRB1 mutations. Amy talked about the advances in animal models. It's very exciting. The FFB is funding the work in this large animal model with pigs that so far it looks like it very closely mirrors that the function of CRB1 in humans. So a lot of exciting things going on there. As Claire mentioned in the Q&A, the FFB takes many shots on goal, and as you heard various panelists talk about. And so supporting the FFB broadly, we don't know where the solution for CRB1 is going to come from. We don't know which path is the best to pursue and what ultimately will be that beacon that we can cling to that will provide a treatment of cure. So we need to cast a wide net and make sure that we're supporting many different efforts. The last thing I would like to say is that these forums will be repeated. It will be something that we do regularly, and we're excited to have you all involved. We hope this continues to grow. I just, again, encourage you to get active, get involved. If you have questions, reach out. And now is the best time for us to really make a difference as a group. And collectively, those of us who are impacted families can make a difference. So thank you for participating, and many more to come. And with that, I'll turn it back to Jason to wrap us up.
I'll take it. Yeah, thank you so much, Jason. And again, it's 12 o'clock here on the East Coast, so we'll wrap up the call. But I want to thank everyone for taking time this Saturday morning to learn about CRB1. As Jason mentioned, and I can't reiterate this enough, this is the beginning of what we are committed to, not only with regards to our financial commitment, but also with our time. We want to make sure that we have frequent updates like we've had today with the CRB1 community. And you can be sure that you can count on that from us. So have a great rest of your day. Have a great weekend, and we'll talk to everyone real soon. Bye-bye.
Bye all.